Hello everybody, this is the Biology Trial 4 exam review for grade 9 at the International School of Innovation. Let's start first with the introduction to genetics, reproduction and genetics. First, what the meaning of reproduction? We see this tree, the tree or the pedigree of the family. We are going to define what is reproduction. So reproduction is defined as the biological process that leads to an increase in the number of individuals in each species to avoid their extinction. So this process is defined by the biological process that leads to the increase in the number of species to avoid what's known by extinction. Note, you have to know that this symbol, it codes for the male, while this other one codes for the female. X, or the cross, means mating. This is a symbol of the cells where they divide to produce new individuals of the same species to avoid their extinction. So reproduction is divided into two parts. Either it's sexual reproduction, which involves the union of the gametes, the male gametes and the female one. Example about this: humans and animals, they undergo what's known by sexual reproduction. We have asexual reproduction, which doesn't involve the union of the gametes, either the male or the female gametes. Example about this: bacteria and some kinds of plants. See, some kinds of plants and bacteria, they undergo what's known by asexual reproduction. Here we move to the gametes. Gametes, they come from the male and the female. Female gametes and the male gametes, here we see the sperm and the ovum. They produce what's known by the zygote. This is the process called fertilization. So what is fertilization? Fertilization, it can be defined by the union of the male and female gametes, meaning that the sperm and the ovum, whenever they meet, they unite, they give what's known by a zygote. So fertilization is the union of one sperm cell with the ovum to produce a zygote. So one sperm plus one ovum produces a zygote. This is sperm plus an ovum. It gives what's known by a zygote. Heredity. What is heredity? Heredity is the transmission of traits from parents to their offspring. This is defined by heredity. Moving to, what is genetics? Genetics is the science that studied heredity. The science of heredity is called genetics. Here we have the cell, an animal cell. Whenever you have to draw any figure in your exam or quiz, you have to draw it using a pencil. You have to label it and you have to give it a title. For example, this title is an animal cell. You have to put it in a box. Don't forget to do this. So where is the genetic information found? This is the big question here. Where is the genetic information found? Scientists, they did an experiment that allowed them to prove what is the genetic information carrier. Where is the genetic information carried? How did the scientists prove that the nucleus is the carrier of genetic information? What did they do? They bring a black mouse, a white mouse, and another gray mouse. What do? They get from these three mice, they extract nucleus from the black, they extract the cell membrane, the cytoplasm without the nucleus, and they inject them or they transplant them inside the gray mouse that's deprived of the nucleus. So they transplant in the gray. What happens here? Results. What happens in this result? The result is we have a black. What happens? This is the analysis here. After injecting the nucleus of the cell of a black mouse into the unnucleated cell, meaning that unnucleated doesn't have a nucleus, of a white mouse, and we transplant them the zygote into the gray mouse. We transplanted the zygote into the gray mouse. A black baby mouse is born. What does this indicates? This is the indication here, analysis plus indication, this interpret. This indicates that genetic information doesn't depend on the cytoplasm or the uterus. It doesn't depend on the cytoplasm of the white, or it doesn't depend on the uterus of the gray, but rather it depends on the nucleus of the cell, which is from the black. That's why we got a black mouse. We deduce here that the nucleus is the carrier of the genetic information. This is the structure of the cell. We have the cell membrane, the centrosome, cytoplasm, nucleus, nucleolus, nuclear membrane, mitochondria, and we have the vacuole. What is the chromosome? Chromosomes are egg-shaped. They look like an egg shape. They are egg-shaped. They are found in the nucleus and they are responsible for carrying the genetic information. So this is the structure of the chromosome. The whole structure is called a chromosome. Each one of these is called short arm, P arm, or the long arm is called the Q arm, and the center is called the centromere. Each one of these is called the sister chromatid. Each one of these is called the sister chromatid. Structure of a chromosome. Each chromosome is made up of DNA, deoxyribose, nucleic acid, and proteins. It's made up of two identical sister chromatids, as we see in the slide before, attached in the center, which is called the centromere. Each chromatid has a short arm and a long arm, a P arm and a Q arm. What is a centromere? It's a constructed region near the center of the human chromosome. It's the attachment site of the two sister chromatids, the site where the two sister chromatids are attached together. This is the centromere. This is the region where the two sister chromatids are attached together. This is P arm, this is the Q arm, long arm, and this is the centromere. Each chromosome carries same genes. Let's see here, hair color gene and hair color genes present in two sister chromatids, as you see, the same position, the same loci. Eye color gene and eye color gene, it's the same position, the same loci. Height gene and the height gene, they are present in the same position. Gene 4, gene 5, G6, and so on. What can we conclude here? What is a gene first? Gene is a part of a chromosome responsible for the appearance of a specific trait. Example about this, hair color gene, eye color gene, and many others. Every gene has a specific locus, specific position of a gene on a specific chromosome. What the meaning of a locus is a specific position of a gene on a specific chromosome. The two sister chromatids of each chromosome, they carry the same gene and the same loci. This is the chromosome. Two sister chromatids, they carry the same genes. If you see here, they carry the same gene and the same loci. So chromosome can be represented only by one. This is genes that are presented on this chromosome. What is an allele? Allele is a version or is a form of a gene. Example, gene of the eye color. We have brown eye allele, we have the green, we have the blue, we have the hazel. Why? The gene of the hair color, we have the black, we have the blonde, we have the red. All of these, they are forms of the gene. They are called alleles. So, types of cells. Moving to the types of cells, we have two types of cells. Either they are somatic cells or they are sex cells or reproductive cells. Somatics, they are diploid cells, while the sex cells or reproductive cells, they are haploid. All body cells except the gametes are somatic, while the gametes, which are the sperm and the ovum, they are sex cells or reproductive cells. Somatic cells, they have two copies of each chromosome, while sex cells, they have one copy of each chromosome. Number of chromosomes is 2n, while the number of chromosomes in the sex cells is 1n. In humans, 2n is equal to 46, while the sex cells, humans, 1n is equal to 23. One copy of each chromosome Here we have 2 copies of each chromosome. Here they are called haploid. Here they are called diploid. Here we have number of chromosomes 1n equal to 4. Here 2n equal to 6 in this figure. In a diploid cell, there are 2 copies of each chromosome. They carry the same genes, the same locus. They are said to be homologous chromosomes. Homologous chromosomes, per 1, per 2, per 3 of a chromosome, they are called homologous chromosomes. Chromosomes of pair 1, we have 2 copies. One copy is a maternal and other is a paternal. So one of these chromosomes is inherited from the mother, it's called maternal, mama, and the other is called inherited from the father, it's called a paternal, papa. These are homologous chromosomes. So note, the pair of chromosomes, they carry the same genes, but may or may not, they carry the same alleles. The 2 sister chromatids of each chromosomes, they are said to be identical, meaning that they carry the same genes and the same alleles. These are identical chromatids, homologous chromosomes. Gene of the eye color, they are present in the same loci. One from the father is called paternal, from the mother is called maternal. Here we have the gene of the eye color. One from the mother is carry the allele which is blue color, while from the father is carry the allele which goes for the green color. So they carry different alleles. They are homologous, not identical. Gene of the level thinking also is present the same loci. Let's have an application here. Consider the gene of the eye color. If we have 2 alleles, green and a brown allele, the possible combination in different persons are brown or brown, both of them, they carry brown allele from the father and brown allele from the mother, or brown green, brown from the mother, green from the father, or green brown, green from the mother, brown from the father, and the last one should be green, green, green from the father, and green from the mother. These are the 4 combinations. Here, this person has 2 identical alleles for the eye color. Whenever we have 2 identical alleles for the same gene, they will have homozygous, or it's called pure, or pure line, pure lace. Okay? While when we have brown and green, either green and brown, this person has 2 different alleles. Different alleles is said to be heterozygous or hybrid for the eye color. Also, green and brown is said to be heterozygous since they have 2 different alleles, and the green is said to be identical alleles. It's said to be homozygous, or it's called pure. When the alleles are identical, the individual is said to be homozygous. It's called pure or homozygous. When the alleles are different, the individual is said to be heterozygous, or it's called hybrid or heterozygous. Dominant alleles. We have 2 types of allele. Either it's dominant or it's recessive. Dominant allele is the allele that will be always expressed in the case of homozygosity or heterozygosity. That's why it's usually symbolized by a capital letter. I'm the king here. I'm symbolized by a big letter since I'm bigger than you. While the other is called recessive. Recessive allele is always expressed in the homozygotes only. That's why it's always expressed in a small letter. I'm a weak one. That's why I'm expressed in a small letter. Considering the gene of the eye color, if the brown allele is dominant over the green allele, complete the following table. We have set of alleles of the eye color and the expressed eye color. Let's see what will happen here. If you have to fill the table, you have to give symbols. Let's B, they said B, brown is dominant. Let B be the symbol of the dominant brown allele over the recessive green allele. So that's why we gave it a capital letter. Why? Let G small be the symbol of the recessive green allele with respect to the dominant brown allele. Set of alleles, either we have BB or BG, or BG, it's called GB From the mother, they differ. Mother, maternal and paternal, they differ here. But whenever we reach the table of cross, table of results, they will look the same. But let's here differ somehow. B from the mother, G from the father. On the opposite is true. And the last one is GG. So BB, B is dominant, so it's brown. BG, B is dominant as you said, and G is recessive, so it's brown. GB, B is dominant, so it's brown also. GG, it's recessive and it's always expressed in the case of homozygosity, that's why it's green. So to say here, this is called the genotypes. These are called the genotype, while the traits that are expressed, they are called the phenotypes. So what is a genotype? According to Freir's model, genotype, this is the definition here. Definition, the set of genes and their corresponding alleles. Example here, BB, BG, GG. Example, a non-example, blue, tall, green, yellow, whatever. This is a non-example. Okay, and this is an example. Moving to definition of phenotype. As Freir's model definition, the set of the observed traits resulting from the expression of genotype. Phenotype, tall, short, black, green, yellow, whatever. This is a non-example. This is for genotype and not for phenotype. For each of the following problems, answer these questions. What would you deduce from this cross? What is the type of this cross? You have to justify. Designate by symbols each of the studied alleles. Perform the essential factorial analysis to verify the results of F1. Perform the essential factorial analysis to predict the results obtained in offspring upon crossing two F1 individuals. Cross 1 or problem 1, pure. Pay attention to here. Pure male black mouse is crossed with pure female female white mouse. Sorry, all F1 generation obtained are black. While in problem 2, a female cat with green eyes is crossed with a male cat having hazel. All F1 they had hazel eyes. So problem 1 solution is first, what can you deduce from this cross? You can say since all F1 generation are black, like one of the parents, then the black allele is dominant over the white allele, which is recessive. What do we call this cross? This is called a monohybrid cross. Why? Since the parents are pure and they differ in only one trait, which is the color. The symbols of the allele, let B capital, be the symbol of the dominant black allele over the white recessive allele. Let W small be the symbol of the white recessive allele with respect to black dominant allele. Number 4, table of cross. Let's phenotype for the parents. This is the male and this is the white is the female. Be careful not to exchange these two, male and female. Male and female should be the same as in the question. Genotype of the parents, this is the male and this is the female. Here we have the gamete should be in the symbols. This is the gametes. Table of cross. 1, this proportion. If you want to use as a percentage, you can use 100% for 100%. Table of results, genotype, it's 1 over 1 BW, so the phenotype is 100% or 1, it is black. Therefore, the results are verified. Moving to self-cross. Whenever we cross F1 cross to F1, it's called a self-cross. F1 crossed by F1, it gives something known by F2 generation. This is the male, this is the female, male BW, female it's also BW. This is the gametes, half B, half W. This is also half B and half W. So we're gonna move to the table of cross and the table of results. BB 1 over 4, BW 1 over 4, also WB or BW as we said in the previous slides. It's the same idea here. So BW quarter plus quarter gonna be half as mentioned here. BB it's gonna be quarter, and WW, which is homozygous recessive, it's 1 over 4. So genotype gonna be BB 1 over 4, BW 1 over 2, and WW 1 over 4. This explains that since B is the dominant allele, So 3 over 4, half plus quarter. 3 over 4, it's black, while 1 over 4, it is white. Moving to a solution of problem 2. Since all F1 had hazel eyes like one of the parents, then the parents are pure and the hazel allele is dominant over the green allele, which is recessive. What do we call this cross? This is called a monohybrid. Why? Because the parents are pure and they differ in only one trait, which is the eye color. Designate by symbols each of the corresponding alleles. Let H capital be the symbol of the dominant hazel allele over the green allele, which is recessive. Let G small be the symbol of the green recessive allele with respect to the hazel dominant allele. So here, let's move to part 4, steps of the factorial analysis. Be careful to these steps. Number 1, you have to write the phenotype of the parents. Number 2, genotype. Number 3, gametes and percentage. Either it's ratio or proportion or ratios, or it's percentage. Gametes, you have to put the symbol of the gametes. Step number 4, table of cross is called Punnett square. And finally, table of results of the offspring, the genotypic and the phenotypic percentages or ratios. Problem number 3. A male Drosophila having gray body was crossed. So this is crossed with the female having a black body. All the F1 offspring, they had a gray body. What we deduce from this cross, what can we deduce is since all F1 has gray body like one of the parent, then the parents are pure and the allele coding for the gray body is dominant over the one coding for the other color, which is the black one. What is the type of this cross? It's a monohybrid cross since the parents are pure and they differ in only one trait. Symbols, let G capital be the symbol of the gray dominant allele over the black allele, which is recessive. Let B small be the symbol of the black allele, which is recessive with respect to the gray dominant allele. That's what we said. You have to make the factorial analysis. Step number 5, this is a new idea for you. When two F1 flies are crossed, so this is called a self-cross. The following F2 offspring are obtained. We have 12 gray flies and 4 black. What is the type of this cross? This is called a self-cross. Calculate the percentage. First step, you have to get the total. 12 plus 14, it is 16. So the property is percentage of x. It's equal to the x over total multiplied by 100. So you have to do this before you can go to the factorial analysis. Step number 6, again the same, but you have to calculate the proportion. We're going to know how to do this. So the solution of the problem is, as we said, since all F1 are gray like one of the parents, then the parents are pure and the gray allele is dominant over the black allele, which is recessive. What is the type of this cross? This is called a monohybrid cross since the parents are pure and they differ in only one trait. Part 3, give a symbols. Let G capital be the symbol of the dominant gray allele over the black recessive allele. Let B small be the symbol of the black recessive allele with respect to the gray dominant allele. Part 4, factorial analysis. You have to know this. Moving to part 5. Part 5 is called a self-cross. How can we calculate percentage? Percentage, this is the general formula. Percentage of x is equal to x over the total times 100. So total is 12 plus 4, 16 flies. So percentage of gray is number of gray over the total times 100. Number of gray, it's a 12. 12 over 16 times 100, it's 0.75 times 100. So percentage of gray, it's 75%. Moving to percentage of black. Percentage of black is number of black over the total times 100. So number of black, it's 4 over the total, it's 16 times 100. So it is 25%, which is the percentage of the black. Moving to part 5, C, self-cross, phenotype gray, genotype GB, 50%, 50%, table of cross and table of results you are familiar with. So you're gonna move to part 6. Whenever you have to calculate the proportion. First step, you have to take the smallest number. We have 12 and 14. Which one is the smallest? Sure, it's 14. It's 4, sorry, 12 and 4. Divide all the numbers by 4. Gray, it's 12 over 4, so 3. Black, 4 over 4, it is 1. So we add the numbers. 3 plus 1, it is 4. So the gray is 3 over 4, and the black is 1 over 4. So moving to activity 2, the laws of heredity. Application: upon crossing a pure line of smooth bees with a pure line of wrinkled bees, all obtained generation of F1 are smooth. So what can we deduce here? That smooth allele is dominant over the wrinkled, which is recessive. Cross 2, upon self-fertilization of F1, meaning that F1 is crossed with F1. We obtain the following: 5 plants with wrinkled seeds, 15 plants with smooth seeds. So application, referring to class 1, determine the dominant and the recessive. Which of Mendel's law is verified? Indicate the genotype of the parents and that of F1. According to class 2, give the gametes produced by F1 plants. To which hereditary laws did you refer? Justify or state them. Calculate the proportion of the different phenotypes obtained. Moving according to the two crosses, perform the essential factorial analysis to verify the results obtained. So in the application 1, cross 1, since F1 P's are smooth like one of the parents, the smooth allele is dominant over the wrinkled allele, which is recessive. The law that is applied here is Mendel's first law of uniformity of all F1 hybrids, meaning that all F1 hybrids are uniform, having the same genotype and phenotype, which is S capital W small. Genotype of parents with smooth seeds, parents with wrinkled seeds, and F1 are SS, WW, and SW respectively. Application here, let S be the symbol of the application note here that S be the symbol of the smooth allele, which is dominant over the recessive wrinkled allele, and let W small be the symbol of the wrinkled recessive allele with respect to the smooth dominant one. Moving to part 2, the phenotype, genotype, gametes. Genotype of F1 is SW, so gametes is going to be 50% S and 50% W. Which law is applied here? Mendel's second and Mendel's third laws. Mendel's second law state that law of independent segregation of alleles, meaning that alleles of F1 separate independently during the formation of the alleles. Third law of purity of gametes, and each gamete carries only one allele for each gene. How to calculate the proportion? As we said before, first step, you have to take the lowest number, which is 5, divide all the numbers by 5. For the smooth, 15 divided by 5, it's 3. For the wrinkled, 5 divided by 5, it's 1. Add these two, it's gonna be 4. So wrinkled, it's 1 over 4, and smooth is 3 over 4. Factorial analysis and the experimental results are verified. Test cross. Test cross is the cross of dominant individual with the recessive individual. It aims to determine if a parent is pure or hybrid. Pay attention to this cross. You are going to have such a question in the exam. Reminder, recessive. Pure means that we have only one phenotype in the offspring. Dominant, hybrid, so we have two phenotypes in the offspring. Okay, pay attention to these notes. These notes are very important for your. Here we go to the types of crosses. This note you have to keep it in your mind. What are the types of crosses? We have the self-cross. Whenever we have, for example, F1 crossed by F1, this is called a self-cross. We have monohybrid cross. The parents are pure and they differ in only one trait. This is a monohybrid. Test cross. We cross an individual with a pure homozygous and recessive. This is called a test cross. This is to identify whether this is pure or it's a hybrid. Cases of heredity, we have a complete dominance where one of the allele is dominant over the other. The second case, which is the important one, and this one you're gonna have a question in the exam about codominance. Codominance means that two alleles they are dominant together. For example, we have a dog which has a white color spotted with a black color. This is codominance, where two alleles they are co-expressed. Intermediate dominance, where there is a color which is intermediate between two colors. For example, in class we solve about the flowers. The flowers they were, for example, white flowers crossed with a red flower. The obtained color is pink. Pink is an intermediate color in the middle between white and red. This is the cases already that we talk about: dominance, codominance, and intermediate dominance. Moving to activity 3, the carriers of the genetic information. What are the carriers? First, let's start with the karyotype. What is the karyotype? Karyotype is the arranged set of chromosomes. Chromosomes, whenever they are arranged in a set, it's called a karyotype. In a diploidal homologous chromosomes, they are arranged as pairs according to the following criteria. In the exam, maybe I'm going to ask you what is the criteria for which the chromosomes are arranged. You have to know that they are arranged according to the decreasing order of size, they are arranged according to the position of the centromere, and they are arranged according to the bending pattern. This is a question, sure you're gonna have a similar one in your exam. This is a karyotype. For example, this is the karyotype showing the different chromosomes. Chromosome number 1, 2, 3, 4, leading to 22. So 22 pairs plus the sex chromosomes here. For example, this is XY. So this goes for a male because we have Y. So this is a male. What are the gonosomes? Gonosomes, they are called also the sex chromosomes. They determine the sex of the individual. Mainly they are X and the Y chromosome. If it shows two X, meaning that XX chromosomes in the gonosomes, so it belongs to a female, to a girl. While if it shows an X and a Y, so XY, so it belongs to a male, which is a boy. Autosomes, they are all the other chromosomes except the sex chromosomes. From pair 1 to pair 22, they are called autosomes. Autosomes, we have 22 pairs of autosomes, numbered as I said from 1 to 22. These are called autosomes. So the gonosomes, they are only the sex chromosomes, meaning that they are X and Y, while the autosomes, they are all the chromosomes starting from pair 1 to pair 22 except the sex chromosomes. Now let's move to activity 4, the chromosomes and the traits of the individual, which means the chromosomal abnormality. Let's start with an application. Sarah and Maher, they are both normal. They have a family history with albinism. What is albinism? Albinism is a disease where melanin is not produced, leading to the lack of pigmentation of hair and skin cells. Sarah and Maher, they gave birth to 4 children: Ahmed, Jack, Mary, and Fatima. All were normal, except Jack, who turned to be an albino, meaning that it has this disease which is albinism. Okay, so construct a pedigree, which is called the family tree also, representing this family. You have to determine the dominant and the recessive allele, designate by symbols each of the corresponding alleles, and you have to determine the genotype of the parents and their children, and you have to perform the essential factor analysis to verify these results. So before we start, we have some notes. Whenever we have a circle, this is a female. Female, this is with trait 1, this is meaning with a normal trait. Whenever we fill it, this is normal trait 2, which is the disease albinism or albino. This is man, which is the box. Box is always for the man. So circle is also for the female, and the box or the square is for the male. This is an empty one, so this is normal, this is trait 1. This is with trait 2, which is with albinism. Whenever we have the geometrical shape, it goes for the fetus, the new baby. Whenever we have a box male with a circle female which are attached together with this line, this means that this is a marriage. Whenever we have two lines, meaning that we have relative mating, meaning that relative between two parents which are relative to each other. They are, for example, cousins or somehow. Plus, we have children where we have the origin and they distribute from the origin. This called the children. And whenever we have this shape, this means we have twins. So we said that Maher and Sarah, they marry and they give birth. So this meaning marriage. They give birth to Ahmad, Fatima, Jack, and Mary. This is the legend here. We have normal woman, which is the circle, which is empty box or empty square for the normal man, and albino man with a box which is filled. So which is the dominant interest? Since Sarah and Maher, they are normal, yet they give birth to an albino Jack, and Jack which has albinism, so the parents must be carrying the albinism allele but masked. Here the key word is masked. You have to know this by the dominant normal allele. So the normal allele is dominant over the recessive, which is the allele of the albinism. Designate by symbols. Let N be the symbol of the normal allele, which is dominant over the recessive allele of albinism. Let A be the symbol of the allele of albinism, which is recessive with respect to the dominant normal allele. Let's say the genotype. Just genotype you have to justify. Whenever I told you give the genotype of something, you have to justify. Usually you have to justify. In the exam mainly I'm going to tell you identify or specify at the rhyme of Wi-Fi, so you have to justify. So since Jack is an albino, albinism is recessive, allele albinism is recessive, so it's only expressed in the case of homozygosity. So that is homozygous recessive with genotype small a, which is AA. Maher and Sarah are normal, yet they gave birth to a albino Jack, so they must be carrying the allele of the albinism, but it was masked by the normal dominant allele. So the genotype should be an A. Since the normal allele is dominant, so it's expressed in the case of homozygote and heterozygote. So Mary, Fatima, and Ahmad could have received two alleles, and from each parent, or one allele and from one of the parents, and one allele A, small a, which call for the albinism from one parent. So they could be pure of genotype NN or hybrid of genotype NA. So you have two possibilities here, either NA or NN. The factorial analysis, NA crossed with NA, normal crossed with the normal. So you have to do the crossing. Let's move to another application. The following pedigree correspond to a family where some members are affected by color blindness disease. This is the pedigree. This one and 2, they marry and they gave birth to 3, 4, and 5. 3 and 4, they are affected, meaning that the allele coding for the disease is masked in the parents. That's why it appears in the offspring, meaning that 1 and 2, they are normal and the allele coding for the disease is recessive with respect to the normal dominant allele. And here we have 5 and 6, also they are normal and they give birth to an affected child, which is 7. This confirms our point of view, which is 5 and 6, they are normal and they give birth to affected child. The allele coding for the disease is masked in the parent and is masked by the normal dominant allele. This explains that the normal allele is dominant over the recessive allele coding for color blindness. Indicate the dominant and the recessive allele and justify your answer. You have to give the symbols of the alleles. Even though maybe in the exam I'm going to ask you what are the dominant and the recessive allele, justify your answer. This part I'm not going to ask you about it. Give the symbols. So automatically you have to give symbols. If I didn't ask you about this, for example, you are going to give the genotype or you are going to do factorial analysis, automatically you have to verify the dominant and the recessive and you have to give the symbols. without asking, you have to give the symbols. If I didn't give you this question, is it made by symbols or give the symbols? Since whenever they use the genotype, from where did you get the symbols? Or from where did you do the factorial analysis? You have to be careful about this. So whenever I ask you about the symbols, you have to write. Did I ask? You have to write. So in both cases, you have to write the symbols, since the symbols are very important to complete the question. Specify the genotype of the members of this family except for the fetus. Specify at the rhyme of Wi-Fi, so you have to justify. Carry out the essential factorial analysis to predict the risk in ratio form for a fetus to be blind. So in the ratio form means that you have to work in proportion and not in percentage. So solution for this application: the normal allele is dominant over the color blindness allele, which is recessive. This is because 1 and 2, they are normal and they gave birth to color blind children 3 and 4. So the parents must be carrying the allele of the disease, but it was masked by their normal allele. Let N capital be the symbol of the normal allele, which is dominant over the recessive color blindness allele. Let C small be the symbol of the color blindness allele, which is recessive with respect to the dominant normal allele. You have to get the genotype. Genotype, they said specify, so we have to justify. So 1 and 2, 5 and 6, they are NC, meaning that they are normal but carrying the allele of the color blindness. Since parents 1 and 2 and 5 and 6 are normal, yet they gave birth to color blind children 3 and 4 and 7 respectively, meaning that 3 and 4 for 1 and 2 and 7 for the other couple, which is 5 and 6. This will mean the word respectively. The first for the first and the second for the second. Then these parents must be carrying the allele coding for the color blindness, but it was masked by the normal dominant allele, so they are hybrids. Genotype for 3, 4 and 7. Since they are colorblind, the allele coding for the color blindness is recessive, so it's only expressed in the case of homozygotes, so they should be pure of genotype CC. Finally, we have to do the factorial analysis. NC crossing by NC, so the gametes they give, and the gametes we have N half, C half. Here we have also this is for the male, let's say. For the female, we have N, which is half, and we have C, which is half. Table of cross, table of results. Let's do table of cross and table of results. So you have to do it using a pencil. This is table of cross. Here we have the male. Here we have the female. For example, this is N half. This is C half. Also, this is N half. This is C half. N 1 over 4, NC 1 over 4, NC also 1 over 4, and CC 1 over 4. Moving to this is the table of cross. Moving to the table of results. This is genotype. This is phenotype. So N 1 over 4. This is NC. We have 1 over 4 plus 1 over 4. It's 1 over 2. It's half. And CC, it's 1 over 4. So N 1 over 4, NC 1 over 2. Both they are normal. So normal 3 over 4. And CC, which is color blindness, which is 1 over 4. So whenever they say to verify the experimental results, you have to say therefore the results are verified. To predict, you are not allowed to say therefore the results are verified. Okay. Moving to this pedigree showing the blood groups of the members of certain family. The gene coding for blood group is found on the chromosome number 9. Chromosome number 9 is it autosome or a gonosome? Sure, it's an autosome. Is the transmission of blood group? This is a question that I was talking about. Autosomal or gonosomal justified, since it's found in chromosome pair number 9, so it's an autosome, so the transmission of the blood group is autosomal and not gonosomal. Among the alleles A, B, O, specify the dominant and the recessive. You have to say schematize the chromosomes and the allele of each individual except for fetus 6. Perform the essential factor analysis to predict. So the results are verified. So starting with the first part, autosomal, since the gene coding for the blood groups found on chromosome pair number 9. Allele A and B are dominant over the allele O, which is recessive. Why? They are co-dominant. Pay attention to the case of co-dominance, okay? Maybe you're gonna have a question like this in the exam. This is because parents are of blood group A and B, and they gave birth to child of blood group O. So the allele coding for blood group O, it was masked by the parents. Okay, so it was masked by the alleles A and B. So the parents should be carrying the allele A and B, so they have the genotype AO, and the other one had the allele BO. That's why they gave birth to child having OO. Moreover, they gave birth to child of blood group AB. So both alleles are co-expressed. Co-expressed meaning that the alleles A and B are co-dominant. So let A be the symbol of the allele, let B be the symbol of the allele coding for blood group A, and let O be the symbol of allele coding for blood group O, which is recessive with respect to A and B. This is the schematized for each of the chromosomes. For example, 1 and 3, they have blood group A, and they have A and they have O, which is mass, so we have AO. Pay attention here, they should be in the same loci, the same region, not here one and the other one is here. For example, they should be in the same loci. Individual 2 carrying blood group B, but they gave birth to a child of having blood group O, so it should be BO. The OO is homozygous recessive, so it should be OO. AB individual 5, since they are co-dominant and they are co-expressed together. So last part, which is the factorial analysis, AO crossed with BO gametes, we have OA 1 over 2, O 1 over 2, B 1 over 2, O 1 over 2. Table of cross results, we have a risk for the fetus to have 1 over 4 chance to be of blood group AB. Moving to the other parts, which is the basic mechanism of sexual reproduction, and we're going to start with the mitosis. So biology is the only science where multiplication and division, they have the same meaning. Multiplication is the cell to divide, and division is the cell to divide. So whenever it's cell to divide or to constrict and give new cells. So the biology is the only science where multiplication and division, they have the same meaning. What is mitosis? Mitosis is the process by which the somatic cells, meaning that diploid cells, 2N cells of the body, they reproduce. They produce new daughter cells. They are identical to the mother cell, conserving the genetic information from generation to generation of the cells. Why would the cell need to reproduce? What is the aim of mitosis? Aim of mitosis is to increase the cellular number and allow the growth of the individual, like in the case of zygote, development into an embryo, or to renew body tissues, replace old, dead, or injured cells. But a cell is not always reproducing. The cell, over its lifespan, will go through the successive life cycle. So what is the life cycle? It's a set of events that the cell undergoes to grow and divide properly. We have divided into two parts, mainly interphase, which includes G1 as G2 gap 1 synthesis phase and G2 gap 2, and the mitosis, which has prophase, metaphase, anaphase, and telophase. So the main life cycle, we have G1 as DNA synthesis and G2 gap 2. This is called, all of this is called the interphase. Whenever we finish the interphase, we have mitosis, prophase, metaphase, anaphase, telophase, and we have the cytokinesis when the cell constricts. So as we said, this is the interphase where we have G1 as G2. G1 is the first gap where the cell is doing its everyday job. The chromosome, they have one strand of DNA. As phase, the synthesis where DNA is replicated here at this step. Chromosomes at G2, second gap, preparation for mitosis. They have two strands of DNA, and we have two mitosis, finally to have two daughter cells identical to the mother cell, having the same genetic information. At the interface, we have the centrosomes, we have the chromatin. We don't have chromosomes, they have chromatins. Inside the chromatin, with the present entire nucleus, this is the nuclear membrane, and this is the chromatin. Here we have the nucleolus. This is the cytoplasm. You have to know how to draw the figures. We have the plasma membrane, nuclear membrane, and the centrosomes. Centrosomes, whenever we finish the interphase, we're gonna move to the mitosis. The centrosomes, they are going to divide and they are giving two asters. They're going to change into asters. We have mitotic spindle fibers. They start to appear. These are the mitotic spindle fibers, as we know how to draw it in the class. This is mitotic spindle fibers, and the chromatin condense into chromosomes. These are the chromosomes, and the nuclear membrane, as you see here, it start to disappear or start to disintegrate. So this is the figure I'm talking about. This is the mitotic spindle fibers, and the chromosomes here are made of two chromatids here, no more chromatin, and the aster, they move to opposite poles, and the spindle fibers are present. So what happens in the prophase? The nuclear membrane disintegrates, meaning that disappear. The mitotic spindle fibers is like the strings. They are formed. The centrosomes, they change into asters, and they move to opposite poles of the cell. The chromatin condense so that individual chromosomes are now individually distinguished, and each chromosome is made up of two chromatids. Mitosis, which is a metaphase. Metaphase, the chromosomes, they are aligned on the metaphase plate, meaning that they are aligned in the middle of the cell. They form something known by the equatorial plate. They form something known by equatorial plate. So this is the prophase. The chromosomes, they line or align at the middle of the cell, the equator of the cell, forming what is known by equatorial plate. Anaphase. What happens in the anaphase? Here we have the mitotic spindle fibers. They pull, each one of these, they pull a part of the chromosome to its pole. So they pull a part of the chromosome to each pole. So each chromosome is made up of two chromatids. They are pulled to opposite pole of the cell, meaning that the chromosome is going to be divided into two chromatids. Each chromatid is going to move to one pole of the cell, as we see here. So what happens? The spindle fibers of the cell pull the sister chromatids of each chromosome apart. Each chromatid moves to the opposite pole of the cell. This process, this is the key word here, is called polar ascension. Wherever each chromatid move to opposite pole of the cell. By late anaphase, the result is we have two identical groups of chromosomes. Each is made up of one chromatid and the opposite poles of the cell. The last step is the telophase. So we have first interphase, then we have, starting with prophase, metaphase, we have anaphase, and finally we have the telophase. So here telophase, we have the cleavage form. The cell start to constrict, the nucleolus forming, and the centrosome, they appear, the chromatin form, and we have the nuclear membrane start to form again. So the chromosomes form two identical groups at the opposite poles of the cell. The chromosomes will decondense. First, at the interphase, they condense to form the chromosomes. Here they decondense to form the chromatin. The nuclear membranes form around each cell, they reform again, and the spindle fibers they disintegrate and the esters return to the centrosome state. The cell membrane constrict, forming what's known by the cleavage furrow. This is the key word here. So remember, at the metaphase we have equatorial plate, metaphase. So we have prophase, we don't have mainly prophase with the chromosomes they are formed, prophase. The metaphase we have equatorial plate, anaphase we have something known by polar ascension, and in the telophase we have known by the cleavage furrow, where the cell constrict to form two cells identical to their parent. You have to keep in mind that the process when the cytoplasm membrane start to constrict, dividing the cell into two identical daughter cells, mainly referred to cytokinesis. Okay, this is the what's known by the mitosis. This is G2 of interphase. This is the starting of the prophase. The esters are formed. We have here the chromosomes. They are consisting each of two chromatin. This is the pro-metaphase before metaphase. Here in the metaphase, they are aligned in the middle, forming equatorial plate. Anaphase, each chromosome is pulled to opposite pole, so each chromatid is moving to pull to opposite poles of the cell, forming what's known by polar ascension. And here we have the cleavage furrow and telophase and cytokinesis, and here the constriction of the cell. Also, this is the mitosis. Mitosis again and again, that's the same process, the same idea, the same concept. We have prophase, metaphase, anaphase, telophase, and we have to go back again: interphase, prophase, metaphase, anaphase, and telophase. You have to study the steps. You have to study what happens in each event, or what are the events in each cycle, in each step: prophase, metaphase, anaphase, and telophase. And you have to know how to draw the figure. Here is also the figure. Maybe you're gonna have a similar figure in the exam. I'm going to ask you about the steps, what happened in this step, what happens. Maybe it's going to be in true or false form. So study well for your exam. Pay attention to the case of codominance. In the case of pedigree codominance, pay attention to what I talk about in class about the different forms of the prophase, metaphase, anaphase, and telophase, how to draw them, how to label them, how to say the difference. Pay attention to, maybe you're gonna have a question either a respiratory system or it's about digestion, one of them. You're gonna have three questions about the genetics, about prophase mitosis, about the pedigree, about the karyotype. So pay attention, study the abnormalities related to karyotypes, related to Klinefelter syndrome, Turner syndrome, trisomy, monosomy. What are the chromosomal formula? You have to know the chromosomal formula of each case. In the normal case, 2n equal 46, 44 plus xy, and so on. Study well for your exam, and good luck, guys.